Hello, welcome to our video. Thanks for joining us. My name is Luciana Burdman. I'm going to present together with my colleague Valeria Casa this work entitled A Journey into Testate Amoeba and Diatoms from Peat Bogs from the End of the World.
Both of us defend our PhD during the last month, and we want to present some of the results we got from working on protists from Tierra del Fuego, Argentina.
Understanding global biodiversity patterns requires taxonomy and biodiversity inventories of comparable quality and coverage depth at global scale. Both of these conditions are best fulfilled for larger organisms, while data are more limited for microbial taxa. Diatoms and testate amoeba are shelled protists, which test are well preserved in the environment, allowing their taxonomic identification and good quality surveys. These microorganisms have been widely studied in peatlands, particularly in the northern hemisphere.
Peatlands represent between 50 to 70% of world wetlands, and the formation of this environment is influenced by climate and also by the topography of the area. That's why most of them occur in distant regions in both hemispheres. Peatlands can mitigate the effect of climate change because they play an important role in the global balance of greenhouse gases, taking CO2 from the atmosphere and dipping it into the soil. In Tierra del Fuego is the southernmost extensive area of peatland, and here we are focused on Rancho Hambre and Andorra Valley. These are two well-preserved ambrotrophic peatlands near Ushuaia city. As part of a major project, our objective is survey diatom and testate amoeba diversity in two Tierra del Fuego peatlands, Rancho Hambre and Andorra Valley.
In order to cover the environmental heterogeneity inside peat bogs, at each of them we randomly selected 4 Sphagnum magellanicum matrix sites, 5 clear ponds, and 4 vegetated ponds. This we did along a transect crossing the peat bogs. And while in the Sphagnum matrix we only find one microhabitat for communities to be in, this is the periphytic associated to Sphagnum magellanicum. In the ponds, we also find periphytic associated to Sphagnum fimbriatum and to Sanionia uncinata, as well as the planktonic and bentonic communities. So all of these samples came to a total of 80. And along with the communities analysis, we made a complete physical and chemical and morphometric characterization of each sampling site.
According to diatom results, a total of 104 diatom species have been identified, belonging to 37 different genera, and most of them were from Eunotia, Pinularia, Forstulia, and also Neidium.
In the case of testate amoeba, a total of 89 species have been identified, belonging to 30 different genera. There is a common pattern between testate amoeba and diatom communities. A large percentage of their diversity do not correspond to any previously described species. This is 55% of the testate amoeba diversity and 53% of the diatoms. While in the case of the diatoms, 22 species were already published as new species. In the case of testate amoeba, we are working on a reevaluation of southern hemisphere diversity, and also molecular analysis was performed for one of the species.
Another common pattern that we found is that from Tistamoeba as well as from diatoms, Rancho Hambre present the greatest riches, and most of the, or almost 50% of the species were in common between both peatlands. And Andorra Valley community was it was a nested ensemble, and Andorra riches is included in the greatest diversity that was observed in Rancho Hambre, from Tistamoeba and also for diatoms. When we analyze an environmental data with a PCA analysis, we can see that Rancho Hambre presents a higher environmental heterogeneity than Andorra Valley, and that could be one of the reasons why it presents a greatest biodiversity also. But we're still working on that. More analysis to come about the relationship with Tistamoeba and diatom community, this diversity, and the relationship with the environmental data.
To conclude, forest diversity in southern South America remains less documented than their northern counterpart, and our data evidence the existence of a high diversity and many previously unknown species in southern peatlands, some of which might be endemic for Tierra del Fuego region.
Finally, we conclude that protist communities can differ between peatlands, even if their overall environmental conditions are similar on eutrophic peat bogs, and the distance between them is short to protist dispersion. We want to thank to the organizer of this meeting, also to our directors and laboratory partners that make it possible, and also all the institutions that make this shop or this research possible. Thank you so much.